Bem, seguindo, eu gostaria primeiro de agradecer à organização do nosso seminário pela honra de conduzir a coordenação da mesa que segue, que será ministrada pelo professor Scott Halstead. Gostaria de chamar. O professor Scott Halstead vai proferir uma plataforma sobre, traduzindo para o português, a Immune Protection Against Dengue Infection Vaccine Performance. Professor Halstead, ontem a doutora Kira apresentou, mas para quem não estava aqui, eu vou repetir um pouco da trajetória do professor Scott Halstead. Doutor em Medicina pela Universidade de Colômbia, atuou em locais como Japão, Tailândia e Coreia, participa de comitês nacionais e internacionais, consultorias, conselhos consultivos e periódicos, contribuiu com mais de 225 artigos científicos publicados em revistas médicas de alto índice de impacto. Nos Estados Unidos, foi cientista-chefe de pesquisa de doenças infecciosas da Anual Medical Research em Bethesda, cientista sênior do Departamento Molecular de Microbiologia e Imunologia da Universidade Johns Hopkins, atuou como diretor de pesquisa orientador sênior na Pediatric Dengue Vaccine Initiative, foi diretor de Ciências da Saúde da Fundação Rockefeller, onde atualmente é consultor e atualmente é professor adjunto, né, da Universidade de Colômbia. Para quem atua na área de dengue, já provavelmente escutou falar na questão do enhancement. O professor Halstead foi a pessoa que desenvolveu essa teoria do enhancement dengue. Então, eu acho que o professor Halstead tem muito a contribuir e hoje trará muitas informações sobre dengue nessa palestra. Professor, please.
Thank you.
Thank you.
Thank you, Dr. Freer. I hope you said I was a troublemaker.
No, don't do that.
Well,
I'm sure not.
I have always been. So I'm going to make more trouble this morning. Yesterday we talked about the miracle of 17D, and there are other miracles, live virus vaccines, that we have been fortunate to use, that you manufacture, some of them. And for years, we have used antibodies as a measure of seroconversion or even a surrogate of protection. But we've never really known how these vaccines protect. We don't know how viruses protect. So it's been a rather amazing experience to have had published now a very large experience with a newly licensed dengue vaccine that showed uneven protection and failure to protect. And from that vaccine, we have learned, I think, a lot, plus a lot of very new, very exciting work that's been published very recently in the last couple of years. So I think our whole paradigm for understanding how viruses protect and how viral vaccines need to be designed to protect is just coming into light. So I'm going to talk about these topics. I'm going to talk about the protection against viruses, but discuss them specifically in the context of the three leading dengue vaccine candidates. So the current dengue vaccines under development, the newly licensed tetravalent vaccine is poorly effective and not safe. And what can we learn about dengue viral vaccine protective immunity from this vaccine? So let's start with the NIH vaccine. This is composed of three more or less whole dengue viruses, 1, 3, and 4, and then a chimera of the dengue 2 here, where they've mixed the non-structural region of dengue 4 and added onto it the structural genes of dengue 2. But otherwise, it's dengue 1, 2, 1, 3, 4. And all they've done is chop out some of the nucleotides at the 3' end, and that has actually damaged the replicative ability of these viruses and converted a fairly normal dengue virus into a weakly reproductive virus that is a vaccine. So this vaccine has undergone almost 20 years of development, and each of the independent viruses, dengue 1, 2, 3, and 4, and then the chimeras, have actually been tested in susceptible human adults, in addition, of course, previously in monkeys and mice and monkeys. So this slow, deliberate process of testing each virus independently characterizes the NIH vaccine. So we know a lot about this vaccine before it was mixed together as a tetravalent vaccine. And these individual vaccines showed 90 to 100% seroconversion. There's very mild reactogenicity. And when the vaccine was put together and a live dengue 2 challenge done 6 months later, there was complete 100% protection. Now, it's very important to understand what is protection when you're challenging somebody with a live virus. Live virus protection really means that the antibodies that are left over from the infection or the vaccination should basically stop the inoculum at its site. Now, the thing that characterizes most of the live viral vaccines we have is that they're injected subcutaneously. Dengue and yellow fever normally are infected subcutaneously or intercutaneously. So to protect, you must kill the virus right then. You can't allow replicative cycles to occur. And in the, we'll mention this a little bit later, but in the preclinical testing of the Sanofi vaccine and many vaccines, people take viremia as evidence of protection or non-protection, but ignore the anamnestic antibody response. So the NIH was smart enough to define protection as no viremia and no anamnestic antibody response. In other words, the inoculum was destroyed at the site. Furthermore, they have a generalized body rash as evidence of the growth of this virus, and there was none of these volunteers who were challenged with dengue 2 experienced a body rash. So that's complete protection, and it's very important to understand what is protection if you're trying to develop an effective vaccine. The other thing about the NIH vaccine is it's been extensively tested by 2 groups, Aravinda de Silva at the University of North Carolina and Daniela Weisskopf at La Jolla Institute for Allergy and Infection. And these are antibody and T-cell experts, and they have studied natural dengue antibodies, natural T-cell responses. And they were able to show that the responses to the NIH vaccine are very similar, both to the attributes of the antibodies that are directed against quaternary virion epitopes and T cells. So I realize that Butantan is not a word that we can use in this auditorium, but that's where it's being made. So now the second vaccine is licensed and being developed by Takeda. This was, of course, developed at the Centers for Disease Control in Fort Collins, and it's based fundamentally on a dengue 2 vaccine, which parenthetically came from my laboratory in Hawaii, had been passaged 53 times in primary dog kidney, and was tested many years ago as part of a vaccine development project at Manguinhos University and then at Aventis. And this vaccine is, remember yesterday I said, well, we always thought yellow fever was the best viral vaccine. And I said, well, no, the Japanese encephalitis SA1414-2 is the best vaccine. And now today I'm telling you, this is the best vaccine. This is a wonderful vaccine, doesn't produce any, I don't know where this rash is coming from, but it doesn't come from this attenuated dengue too. So this is, all we did in Hawaii was just pass it and pass it and pass it. Nothing fancy like Max Tyler did, which he went from one species to another. So at any rate, the CDC chose this vaccine as their backbone and then inserted the genes for the structural genes for dengue 1, 3, and 4 into this backbone. So the NIH vaccine is essentially 3 whole live viruses with the C prime end attenuated. This is a vaccine where 3 out of the 4 viruses are chimeras of dengue 2, sorry. This vaccine has moved along in preclinical testing and in phase 1 and 2 clinical trials in the United States and various countries in Latin America. And it's now in extensive phase 3 clinical trials in a number of Southeast Asian and American dengue endemic countries. Now, the vaccine from which we have learned a lot is the Sanofi Pasteur vaccine. And you recall that this is entirely a chimera based on 17D, where the structural genes for dengue 1, 2, 3, and 4 are inserted into the backbone of yellow fever. So you have C and the non-structural genes from yellow fever. So the virus, the virion as it's composed, of course, has dengue envelope proteins and PRM. But when this was tested in 35,000, which includes placebo, a 2-to-1 ratio of vaccine to placebo subjects, the overall vaccine efficacy was disappointingly low, at least based on traditional outcomes: measles, mumps, rubella, varicella, yellow fever, Japanese encephalitis, all are in the high 90s. Japanese encephalitis vaccine was 99.4 protective. So what's going on? Well, it turns out there are a lot of children who were vaccinated who were hospitalized with a breakthrough dengue infection. And in all ages over a 5-year period of reporting, this is the 295 occurred in a group of 20,000 children who were vaccinated, resulting in 1.44% of all the vaccinated children developed a dengue infection, which resulted in hospitalization. That figure in itself is remarkable because a child born in any Southeast Asian country today has a lifetime expectancy of being hospitalized for dengue of 1%. So in 5 years, the hospitalization rate in this vaccinated group already exceeded the lifetime expectancy. And you see that if you just looked at the children who were vaccinated as 2 to 5-year-olds, at the 5th year, 2.8% of those children, that's a small group, but nonetheless, a very highly significant fraction of infected, vaccinated children are sick enough to be hospitalized. Now, what happened? Well, the first thing you have to understand about dengue is this is not yellow fever and it's not measles. This is a disease where there are 4 different viruses. You've got to have a vaccine that's going to protect against all 4. You're vaccinating a very diverse group of individuals. Some of them are seronegative. Some of them have had 1 single dengue infection. Some have had 2 or more. And in some countries, the immune responses are complicated by the fact that the children have either experienced a wild-type Japanese encephalitis infection or been vaccinated. For example, in Thailand, Japanese encephalitis vaccine is mandatory, so every child at age 1 is given 3 doses of the old killed vaccine. Yellow fever will be the same thing here in Latin America. West Nile, we have West Nile circulating in many countries in the Americas, and now Zika. So all of these flaviviruses can add complexity to both the immune response, the protection afforded by the immune response of these different vaccines. So we only have 2 pieces of data from the Sanofi published papers, of which there are many, that are actually insightful into what is the mechanism of protection or failure of this vaccine. And this is one of the bits of data here. It's vaccine efficacy. Now I said 1 year post 3rd dose. If you read the paper, they call it 2 years. It is 2 years from the first dose given, but it's 1 year. The Sanofi vaccine schedule takes 12 months, so 0, 6, and 12 months to get 3 doses. So this is 1 year following the completion of the 3rd dose in people who were shown to be seronegative. Now, it turns out that only 12% of all the 35,000 children were bled prior to being vaccinated. So this data is based on children in that 12%. There are not very many of those children, and that, of course, lowers the statistical significance of the observations made. So you can see that even though there was modest protection of seronegatives, the confidence limits went below 0, and the p-values are all insignificant. So this, you have to say that we don't know what the protective efficacy in seronegatives is. It's poor, and basically you can say it's not statistically significant. So there is little or no protective efficacy in seronegatives. Now, seropositives, who are that other group that I mentioned there, monotypic immunes, multitypic immunes, and possibly even people, for example, if you look at the paper from Southeast Asia, the Coppeding paper published in Lancet, when they characterized the seropositives, they're including Japanese encephalitis immunes, which 20% of those children were immune to Japanese encephalitis and not dengue at all. So this is quite a mix, flavivirus mix here. At any rate, the efficacy is considerably higher. You can see almost 80%, and the p-values are very high. The confidence limits look good. So this is a vaccine that protects people that have had 1 or more dengue infections. You get a booster effect, and don't forget that in the real world, it's all the people that have had 1 dengue infection that are at risk of being hospitalized with a 2nd dengue infection. So you are protecting that group. And this is a measure of the serological response to 3 doses of vaccine. You can see, measured 28 days after the 3rd dose, that almost all of these children developed neutralizing antibodies to all 4 dengue viruses. So we learned from this already that the presence of neutralizing antibodies in dengue is not a surrogate for protection, because these children, despite the fact they all had tetravalent neutralizing antibody responses, still had breakthrough dengue infections. So one of the things that Sanofi concluded was that children over the age of 9 years and older were more largely protected, and children under were at a higher risk. And I just wanted to point out that the broad neutralizing antibodies that Sanofi had raised, that I just showed on the previous slide, rather resemble the antibodies that we see in dengue, and what really is one of the most remarkable biological clinical phenomena in virology that I know of, and that is that mothers who transfer antibodies that look like this, broadly protective neutralizing antibodies to their infants, that infant, a few months later, loses those antibodies to a point where the antibody is at an enhancing concentration, and the baby becomes a patient with dengue shock syndrome. This is a 6-month-old Vietnamese girl, and this is not, for some reason, you're not seeing very much of this in Brazil, but I predict that sooner or later it'll occur, because if you have mothers that are highly immune, it's those antibodies that wane. And here's the age distribution. You can see very few cases in the first few months of life. So the maternal antibody initially is protecting these infants, and then it's catabolized. We know that the half-life of human serum IgG is about 40 days. So every 40 days you're diluting 1 in 2, 1 in 4, 1 in 8. So by the end of the first year of life, you're way out to a very high dilution. And so as you have 100% of antibody here, they're protective, it wanes, you wane through a whole zone of enhancement, and then finally, even the enhancing antibodies are gone. So these antibodies turn out to be 10 times more capable of causing hospitalization. In other words, these infants are hospitalized at 100 per thousand, as opposed to older children who are hospitalized at a rate of about 20 to 30 per thousand. So the whole point is that these broadly cross-reactive, non-neutralizing antibodies are actually quite efficient in producing enhanced disease. And this is, for those of you who don't work with enhancement, this is what an enhancement curve looks like in vitro. This would be a monocyte or macrophage culture or continuous Fc receptor line. And over on the left-hand side is undiluted serum, and as you dilute the serum, you go through a period where the virus is neutralized, and then you get to a point where there's more virus produced than you put into the culture, and it peaks and then it wanes. So these curves look alike, don't they? This is a curve in vivo, and this is the curve in vitro. So there are actually two types of antibody-dependent enhancement. One that we call extrinsic, which is just reflects the efficiency with which an infectious immune complex can get onto the surface and into the internal environment of a Fc receptor-bearing cell. And it's about a threefold increase. But there's another phenomenon which is really quite amazing and curious, and that is that a dengue immune complex sends a signal down the Fc receptor to a tail, which actually suppresses type 1 interferon or induces the production of IL-10. And here's the difference between extrinsic. All of you guys in the lab that are using K5 F6-2 cells, all you're measuring is extrinsic ADE, because these are interferon deficient cells, and you've got to have interferon present in order to suppress it to express the intrinsic But this is the important thing. If you want to study ADE, you really want to pay some attention to what's going on inside the cell to increase dengue productivity. So turns out that leishmania, that Kala-Azar, is actually a form of antibody-dependent enhancement. And what happens is you get the amastigote in the macrophage. The macrophage is the obligatory host cell for the amastigote. And as soon as you start making IgG antibodies, the immune complex actually suppresses the immune elimination, and then Kala-Azar turns into an inevitably fatal disease. So there are many illustrations in veterinary medicine. This is dengue ADE is not restricted in biology to just dengue. It occurs in many organisms and many species. So this is the two types. So here's the interferon type alpha and interferon suppression system over here, and here's the IL-10 production system here. So I mentioned before that the infants have a higher rate of hospitalization with passive antibody than this active secondary infection. So here we're measuring the rate at which 2 to 5 year old seronegative children given the Sanofi vaccine are hospitalized. We start off with 20 hospitalizations in this age group and a total of 2,000 vaccinated children and 2 in the 1,005 placebo controls. And we know what percent of seronegatives existed in the 2 to 5 year olds because Couteville published an amazing paper with an 80-page appendix, which had seroprevalence and virus infection rates and multitypic and monotypic neutralizing antibodies for all of the 10 sites in the Sanofi clinical trial. So these are actually real data. So we could estimate 1,100 seronegatives. The infection rate is from the Couteville paper. And so we can estimate 176 seronegative children were infected. And of those, 20 were hospitalized for 11.4%. Now, if we look at the placebo group, there's 1,005 percent monotypic immunes. It's only the monotypic immunes and the placebos that are at risk of getting an infection that hospitalizes them. So there are 379 monotypic immunes, and 16% of which are 61, 2 hospitalized, and the hospitalization rate is 3.3% for natural secondary dengue infection, as opposed to 11% for the children who were vaccinated. So the vaccine makes it 3.5 times more likely that you're going to have a disease severe enough to be hospitalized. So the infants and the vaccinated children, all of who have these broad cross-reactive but non-protective neutralizing antibodies, are actually efficiently enhancing in this case, it's the child's first dengue infection. So what about children over the age of 9? Now, under 9, we can see quite clearly that the relative risk in children vaccinated in these age groups was 5. But that actually mirrors what we know from real life. These are data from Cuba, where in 1981 there was a hospitalization rate. Sorry, these are the data from Cuba, where we were able to estimate what the age-specific hospitalization rate per 10,000 secondary dengue infections was. You can see that the youngest children, aged 3 and 4 and 5 and 9, had a much higher hospitalization rate than did other children. But although the numbers fell, they don't disappear. They once you get to be a late teenager, your susceptibility to vascular permeability plateaus. So what we're seeing here in the Sanofi trial is that the children in this very high age-specific susceptibility to vascular permeability are the same ones that are being hospitalized with a dengue infection after being vaccinated. And in the older age groups, the rate is lower. So the two curves look very similar, and our contention is that there's nothing new about the response to Sanofi vaccine. It is creating an immune response, which then couples with the incoming wild-type dengue infection to produce a disease of the same severity in the same age groups. And this is all based on the published understanding that capillary integrity is much weaker in the very young. This is a chart of, so up here high means you have weak capillaries that tend to leak, and down here you have stronger capillaries. So when you're very young, your capillary system is more tender, if you will, and so the stimuli which cause vascular permeability in dengue are more effective in the younger children and less effective in older children. So the other thing about the cutoff of age 9 is that these are data from Thailand showing the actual age distribution of children admitted to Bangkok Children's Hospital, now called the Queen Sirikit Institute, for a 5-year period. And you can see that the rate continually climbs and peaks at 9 or 10, and then it begins to fall off. And what this is reflecting is that as a child is born, this child is susceptible and then gets one dengue infection and ultimately gets 2 dengue infections and so on. And what this is showing is that 9 is the peak age which children in natural settings have the most percentage of monotypic immunes. And following that, as they live longer, the percentage of monotypic immunes falls off. So the whole business of 9 is just an actual demonstration that in Asia, that is the peak age of susceptibility, the peak age of monotypic immunes. And as you get older, there are fewer and fewer monotypic immunes. You know, if you've had 2 or more dengue infections, you're not at risk to clinical disease. So 9 is truly not a biological age landmark. It's an epidemiological landmark based on the epidemiology of dengue in Southeast Asia. You in Brazil probably have a different peak age admission to the hospital because it all reflects the average annual force of infection or average annual dengue infection rate. So here's the distribution of age and placebos in the Sanofi trial. You can see that it's exactly like this one. It's always reassuring to see that the data confirm what we observe from other. Now, why doesn't dengue vaccine protect seronegatives? One dose of 17D raises antibodies to presumably to quaternary epitopes on the neutralizing epitopes. And you can see that it's been demonstrated that yellow fever antibodies directed at E do not neutralize yellow fever virus. Instead, neutralization is mediated by antibodies to quaternary epitopes on the complete virion. So if you just make a vaccine with E, it just express E protein, you'll develop antibodies to E, but you won't neutralize yellow fever virus. So it requires an antibody that actually fits into a groove on the virion surface. So does Dengvaxia raise antibodies to quaternary epitopes? Well, we have some mixed messages here. We have a paper by Heinen, just published a few weeks ago in JID, showing that indeed there are a significant number of patients who've received the Sanofi vaccine who develop dengue 4, and these dengue 4, by an absorption process, can be shown to actually attach to these virion surface quaternary epitopes. So 7 out of 11. But the antibodies directed against dengue 1 through 3, that were very few of those were actually quaternary epitope antibodies. Now the protection rate against dengue 4 in all of the clinical trials was around 80%. So it may be that this quaternary epitope dengue 4 antibodies are indeed somewhat protective, but you'll see there's more to the story. So remember how Chimera-Vax is structured. That is that we're only looking at producing antibodies to the envelope, or even the virion conformation envelope, but all of the immune to non-structural genes is to yellow fever. And how does this affect the T cell response? Well, we have an amazing number of studies now. One of the nice things about dengue is that people can get dengue all over the world. We can prove that they have dengue, they survive, and you can go out and bleed them. For yellow fever, basically all we can do is measure the immune response to yellow fever vaccine. There's very little equivalent T-cell work done on people who survive virulent wild-type yellow fever. So we have a lot of data for dengue, and the key to good T-cell data is you've got to use the T-cell, the HLA restriction, in order to pick out the correct T-cell epitopes. So HLA dengue resistance studies have shown that human CD8 T cells protect against severe secondary dengue infection, and human CD4 plus and CD8 plus T cells responses in the NIH vaccine are identical to the responses following dengue or infection. And if you haven't read this T cell work, I've listed the authors here. You can see there's a lot. Very, very recent stuff. There's 2 publications in PNAS, and there's some, for those of you who are interested just in summary papers, there's some very good summaries in Frontiers of Immunology in 2014 and 2016. But this is basically what Dr. Weisskopf has discovered, and that is that when you map out T-cell epitopes using the HLA restriction, and then here you've got to make generalizations across a broad number of different population HLAs, but using those restrictions, and she was able to show that the dominant T-cells, dominant antigens recognized by dengue T-cells are in the NS3, NS4B, and NS5. This is for CD8. And for CD4, interestingly, it's CD4 are really directed a lot towards C, which in the Chimera-Vax vaccine is yellow fever, and then NS2A and NS3. So the point is that both the CD8 and the CD4 T cells are directed against epitopes that are on yellow fever proteins and not dengue proteins. So the other thing that's interesting is that each of the 4 dengue viruses has a different pattern of T-cell stimulation. So dengue 1 and 2 and 3 and 4 are all a little bit different. You can almost tell which virus you're dealing with because of the unique dengue type specific pattern. So the lessons learned from natural infections are that the protein location of T-cell targets are really critical, and the responses against non-structural protein targets are dominant. Immune dominance is influenced by the serotype, and of course there's HLA restriction of the target T-cells. So the conclusions are that there's a different immunodominant pattern for class 1 CD8 and class 2 CD4 responses. And C is the main target for class 2. This has implications for mechanisms of T-cell help. The CD4 megapool allows study responses in small samples irrespective of HLA restriction and serotypes. And the NIH vaccine induces CD4 T-cell responses. NIH vaccine responses are higher than those observed in natural infection. The immunodominance for CD4 NIH vaccine differs from CD8, resembles the neutralization index for CD4, and a similar pattern of HLA dominance. CD4 epitopes target in dengue 2 live attenuated vaccine are comparable to natural infection. They actually tested a group of human volunteers that were just infected with dengue 2 and to look at the CD4 epitopes. And the NIH vaccine immunization induces balanced serotype responses. So what is the inability of dengue vaccine to protect seronegatives taught us? The most likely explanation is the absence of a fully competent CD4 and CD8 T-cell response. Why did this vaccine, which made neutralizing antibody to all 4 types, why did it fail to protect seronegatives? You can see that basically the CD4 and CD8 T-cell responses were largely directed against yellow fever epitopes, and there's no significant cross-protection between yellow fever and dengue. And T-cell immunity seems essential to long-lasting protection by viral vaccines. T-cell immunity requires the presentation of the whole replicating virus. Now let's remember, let's go back to what I said at the beginning. We've had a lot of miracles in the field of vaccinology, and here are some of them: measles, mumps, Japanese encephalitis, rubella, smallpox, varicella, yellow fever. These are all whole virus vaccines that have been attenuated by a completely mysterious, most of them by a completely mysterious process of selection. And as the years have gone on, we've seen that they pretty much work. We now have everybody agreeing that yellow fever is a lifetime 1-dose vaccine that looks like the Japanese encephalitis. Measles is pretty good, and so on. So now people will say, yeah, but we have killed vaccines. We have killed Japanese encephalitis vaccine. We have killed polio vaccine. We have killed tick-borne encephalitis vaccine. Well, the interesting thing about these kill vaccines is that these are diseases where, again, virus, polio is enteric, and the others are mosquito or insect transmitted. But there's a period of viremia that precedes the invasion of the central nervous system. And that takes about 5 to 7 days. So if you have a viremia and you're sensitized to produce antibodies, that gives you enough time to mount an antibody response that may actually protect the central nervous system, the spinal cord for polio, and the brain for TBE or Japanese encephalitis. So there may be a perfectly rational explanation why killed virus vaccines protect this type of a disease with what I call a secondary target. But, you know, in dengue, you don't have any time. Zika, you don't have any time. The mosquito bites, and within 5 days, you have a fever. So I think we're not going to find that killed vaccines are very useful. So that's my talk. I know that a lot of people in the audience are possibly not going to be happy.
Thank you, Professor. I think we have time to have a question. If you prefer, take a seat, please. Well, we are...
Okay, thank you. Sandra Besada from Sanofi Pasteur.
Yeah, thank you for... I will ask everybody to introduce yourself.
Okay. I want to give some comments. First of all, regarding the cellular response. Nevertheless, the higher cellular response is against NS3 protein. We evaluate the cellular response for our vaccine, and we have several papers where we show that we have cellular response. We activate CD4 and CD8 against envelope protein, and the pattern of response is protective. So we increase the interferon higher than the necrosis tumoral factor. So our vaccine does not produce rash, for example, that is related with TH2 response that is not protective. First. Second, regarding the clinical development, we follow the WHO guidelines. So we have active phase, that is 2 years of follow-up, just to show the efficacy and also the safety of the vaccine. And we have also 4 additional years of follow-up in order to have 6 years of long-term follow-up in order to evaluate the safety pattern of the vaccine. We already finished 6 years of follow-up of this phase 2B study, the CYD 2357, done in Raspberry, Thailand. And we presented the data in ACT Congress in Asia in 2016. And after 6 years of follow-up in the target population, that is the population older than 9 years old, we have a relative risk of 0.4 with a narrow interval of confidence. That means that is statistical significance, so the vaccine didn't show any risk in population older than 9. In population younger than 9, the relative risk was 0.9 with a broad interval of confidence. Regarding the CYD14 study, the phase 3 study done in Raspberry, Thailand, in around 10,000 subjects from 2 to 14 years, we already have 5 years of follow-up. We published that 1 week ago in Bethesda as a poster. Here I have some copies if you want to see the data on detail. And in population older than 9, the relative risk is again 0.4 with a narrow interval of confidence from 0.2 to 0.6. That means that is statistically significant. That means that the vaccine does not increase the risk in the target population. And in the younger population, younger than 9, the relative risk is decreasing. During the year 3 was 1.4 and now is 1 with a high broad interval of confidence that is not significant.
Okay, can you conclude?
In conclusion, in the target population until now, we have several data from Asia and also from Latin America, and we haven't shown an increased risk of hospitalization in population older than 9, and in population younger than 9, the relative risk is decreasing.
Thank you. Dr. Professor Halstead, do you want to comment something?
Well, I gather you don't think that you don't agree with these data. All I can say is that T-cell immunology is very difficult, and we've, in the dengue field, we've had a lot of T-cell work done over the last 20 or 30 years that actually hasn't ended up teaching us very much. The problem with T-cells is that virtually any antigen you can think of will elicit T-cell responses, and if you use a standard interferon or cytokine response, you have a sensitized T-cell and you expose it to an antigen, you measure interferon or a cytokine. That isn't the same thing as measuring T-cell killing. So measuring T-cell killing does require HLA restriction. So it's much more difficult. There are many fewer studies in the literature that are actually focused specifically on identified T-cell killing. But the thing about dengue, and I could only hope that the yellow fever people had the same access, is that we actually have now a significant amount of T-cell data from human beings that have actually been infected with wild-type strains and that have gone on to experience a disease or no disease. So we can begin to base both the T-cell epitopes that I showed you and the contribution of T-cell to protection on actual wild-type infections in human beings. So having that information then allows you to transfer that information to the different vaccines. So I think in that standpoint, dengue is quite lucky. Since I've been reading the T-cell papers written by this group from La Jolla, I've started to look at what do we know about T-cells and measles, what do we know about T-cells and mumps and rubella and so on. Well, it turns out not much because it's difficult. So T-cell is, I'm sorry, has been sort of the undercurrent in the protection game that difficult to measure and therefore somewhat underestimated. And we've all, I belong to the same generation as all the rest of you guys. We've all seen that you can passively transfer antibody. Antibody will protect and so on. But we shouldn't confuse short term. These babies are protected. But if you don't have some mechanism to sustain that protective level of antibody for years and years and years, it isn't going to work. So it looks like what takes place in real life is there's an antibody response that maybe all by itself is protective, but it's supplemented by a T-cell response, and those T-cell responses are really critical.
Okay, thank you, Hans. I think we are very delayed, but I have space for 2 questions. Elena, please. 1? Just 1? Okay.
Hello. Yes. Thank you, Dr. Halstead, for your lecture. It's an honor to have you here. And I have a question. Considering some of your results that vaccine-induced neutralizing antibodies, but these neutralizing antibodies does not protect individuals, do you think there is a minimal level for these neutralizing antibody titers to be considered protective, or our PRNT are not measuring neutralizing antibodies? We need new testings to prove the efficacy of dengue vaccines?
Yeah, no, I think that's, I don't know how many people have ever read the papers. There's so few of them, of these infants. It's really such a remarkable biological phenomenon. And I've always felt that the people who work on dengue don't like to study infants because it's awkward, it's embarrassing, you know, gosh, this is awful. Here's a mother giving antibody to the baby. So it isn't very well studied, but it's something very important because you can see there that the antibodies at birth are very protective. And there have been a number of studies measuring the different antibodies for different mothers. And some of the mothers will have antibodies that will protect out for 9 months. So we know that in real life, that is in humans, and you can duplicate this in animals, that high concentrations of antibodies, specifically if they, I hope you're all following the work of Aravinda de Silva and others who are showing by process of absorption that you can actually identify that the very small percent, maybe 1% of all the antibodies that are circulating are neutralizing, and they're the ones that actually attach to the virion surface, to these quaternary epitopes. So it looks like the antibodies that are really biologically effective are the quaternary epitopes. They're a very small percentage of all the antibodies that are circulating. And if you know, if you've watched Ted Pearson's work and others, you know, there are 90 of these quaternary epitopes on the surface, and neutralization requires that all 90 are covered. So you have to have fairly high concentrations of antibody to get neutralization. You get down to 89 or 70 or something, and then all of a sudden the virus is no longer neutralized because there's a certain segment of the virus that still has a receptor available for entry into the cell. So I think neutralization is a numbers game. And when you're making a vaccine, you've got to be very careful to make sure you're not falling into the trap that says high concentrations of antibody protective. You know, if you just look at what poor old Tom Monath did, he immunized monkeys and then challenged them 3 weeks or 6 weeks later. You can't do that. You have to wait for the antibodies to fall below some titer that they're going to fall below. And then if you challenge, so that's why the NIH challenge was done at 6 months. So remember, antibodies, yes, antibodies are protective. And we have all thought for years and years and years that was the end of the story, but I think really it's not the end of the story. And yeah, if we go forward now with new vaccines, and I hope we can, we've got to make sure. There may be a perfectly good way to get T-cell epitopes and just put them together with your B-cell epitopes. I haven't seen anybody design a vaccine specifically to do that, but there's no reason why it couldn't. You know, I keep hearing rumors about DNA vaccines. Well, that's fine. Yeah. If you bring in enough of the virion in a DNA vaccine so that you give the immune response a chance to build protective T-cell immunity, that's fine. So I think we're finally possibly headed in a really constructive direction. But if you haven't read these papers by the Weisskopf group, please do.
Thank you, Halstead. I think you can conclude that develop a dengue vaccine still be a big challenge, and a good vaccine we need, but it will maybe take time to have a perfect vaccine. You should have a lot to do. Thank you, Professor. Okay. Thank you.